It's 1953, Upper Nile Basin, South Sudan, a place so hot the air itself seems to press down on you. A British anthropologist named D.F. Roberts unfolds a measuring tape beside a Dinka man near the settlement of Rumbek and writes a number into his field notebook. Not the tallest man he's measured, not the outlier, the average. 182.6 centimeters, the average Dinka man. For context, the global average for men today sits around 171 centimeters. Roberts wasn't measuring extraordinary individuals. He was measuring ordinary ones. That number would circulate through physical anthropology for the next 70 years. And if you've ever read anything about the Dinka, about the people of South Sudan, about the Nile Valley, about extreme human physical variation, you've probably heard the explanation that goes with it. Allen's rule, longer limbs in hot climates, heat dissipation. The body using its geometry to cool itself. The Sud floodplains of the Upper Nile are punishing. Temperatures push past 4 degrees Celsius. A lean body, long legs, long arms. It's a radiator. It works. The logic is clean, so here it is. The explanation you were probably expecting. Ancient climate selected for a body type. The Upper Nile is hot. Taller, leaner bodies handle heat better. Generations of selection pressure lock that body type in. The Dinka are among the tallest people on Earth because of where they live. Case closed. That explanation is not wrong. It is also embarrassingly incomplete. In 2015, researchers published a genome-wide analysis of 232,684 individuals, children and adults, drawn from 80 different ethno-linguistic groups across 3 continents. They were trying to establish exactly how much of the relationship between body proportions and climate is driven by genetics versus by a living body responding to its current environment. What they found was this: genetic population structure, not current climate, not current diet, not current living conditions, accounts for between 50 and 74% of all variation in body surface to volume ratio across human populations. The Dinka's body is not primarily a response to today's heat. It is a record of something that happened to their ancestors. Written in bone, passed down for hundreds of generations, the heat pointed the direction. Genetics locked in the magnitude, and then locked the door. And here's where it gets strange. Because the same genome that produced the world's tallest average human body type turns out to be the genome that modern science treats as something else entirely. Not a subject, not a curiosity, something closer to a fixed point. If you've ever actually wondered where you come from, not the family story your grandparents told, but the real biological answer, tracing your DNA back thousands of years, then subscribe. Because what comes next is not the explanation. It's the question underneath the explanation. And it goes deeper than most people are willing to follow. Before we go any further, I want to know something. Drop it in the comments. Where does your family come from? What region of the world do you think your ancient ancestors called home? One sentence. I read them all. And I mean it, because by the end of this video, your answer might look different than it does right now. 50,000 years ago, the human story was still being written in a single place, Africa. A continent of populations moving and mixing and diverging, some lineages branching away from others, some persisting in one place for so long that the rest of humanity's genetic tree kept splitting around them. 50,000 years ago, the Dinka's ancestors were already living along the Nile corridor, and they were already starting to become distinct. What makes the Dinka genome remarkable, really remarkable in a way that has nothing to do with height, is this. When geneticists at Harvard's Department of Genetics want to reconstruct the ancestry of ancient Africans from 50,000 years ago, they need fixed reference points. 3 populations whose genomes define the genetic space in which all ancient individuals will be plotted. Think of it like a coordinate system. You need known, stable positions to locate anything else. In 2022, Dr. Mark Lipson, Dr. David Reich, and their colleagues published what became one of the most important papers in African archaeogenomics of the decade. It appeared in Nature. It analyzed the oldest sub-Saharan African ancient DNA ever recovered, including individuals who died 18,000 years ago. To run that entire analysis, to place every single ancient individual in their correct position in human genetic history, the research team selected 3 anchor populations: the Judawansi San of southern Africa, the Mbuti of Central Africa, and the Dinka of South Sudan. The Dinka don't appear in the conclusions of that paper. They appear in the methods. That is where the fixed points live. And this wasn't a one-off. The Dinka genome functions as a reference anchor in multiple landmark studies. In Prendergast and colleagues' 2019 science paper, in Wang and colleagues' 2020 Science Advances paper, some of the most cited archaeogenomic research of the past decade, published in the 3 most prestigious journals in the world. Every time the same function, not the subject being explained, the instrument doing the explaining. This discovery was confirmed across independent research teams across a decade of studies. Almost nobody outside the field talks about it, until now, because here's what that means. A reference coordinate in genetics is not a subject. It is not something being measured. When geneticists want to know where an ancient Kenyan herdsman from 3,000 years ago fits in the human story, they calculate his genetic distance from the Dinka. When they want to know where a prehistoric pastoralist in Tanzania sits in the genetic landscape, they measure his ancestry relative to the Dinka. The Dinka are not the thing being described. They are the ruler. Everything else is what gets measured. And to understand why this particular population in this particular place ended up performing that function in modern science, you have to understand what happened to the populations around them. Because not everyone survived, and the ones who didn't leave a name. Every population that lived along the wave left no name, no language, no descendants. That's not a metaphor. That's what the DNA shows. Approximately 2,300 years ago, an individual was buried at a site in Kenya now called Hyrax Hill. He had iron tools, ochre marking, the material culture of a specific functioning community. Researchers would recover his petrous bone, the densest part of the skull, the best preserver of ancient DNA, and sequence his genome. His ancestry broke down as follows: 33% from a Dinka-related source, 50% from a Pastoral Neolithic population, and the rest from another group. His genetic profile doesn't match any living population in Eastern Africa, not one. He is, in the language of genomics, a dead end. His people don't have descendants that anyone has found. His name was never recorded. His language doesn't survive. He appears in the scientific literature only because his bone preserved enough DNA to sequence, and only because the Dinka genome existed to serve as the reference point against which his ancestry could even be calculated. Dr. K. Wang, working with Dr. David Reich and colleagues at Harvard and the Max Planck Institute in Leipzig, published this analysis in Science Advances in 2020. What their study documented was a Nilotic-related population wave moving through Eastern Africa between 3,500 and 1,500 years ago. Populations in its path were not simply absorbed into it, they were replaced. The individuals carrying the pre-expansion ancestry appear in the ancient genomic record, and then they stop appearing. The wave that the Dinka's ancestors rode is the wave that made those people ghosts. But here's where it gets strange, because the standard story of the Nilotic peoples tends to emphasize something different: cultural continuity, deep roots, a people steadily moving south with their cattle. And that story is true. What Wang and colleagues added to it is the darker counterpart. That continuity was built, in part, against a background of populations who cannot be named, whose children did not survive to carry their genes forward. The Dinka genome preserves the record of what expanded. It does not preserve the record of what was lost. And this is where you have to ask the question the field has been sitting with quietly for years. What specific property of the Dinka genome allowed it to survive intact, while everything around it was either absorbed or replaced? What was the mechanism? It wasn't just geography. It wasn't just luck. Something in the structure of Dinka society, something encoded in their way of living, preserved that genome so completely that 50 years from now, geneticists are still going to be using it as a fixed reference point. And the answer turns out to live inside their cattle economy. The mechanism is not in the Y-chromosome, it's in the marriage. Researchers working across Uppsala University in Sweden and the University of Khartoum in Sudan published a comprehensive genomic study of Northeast African populations in PLOS Genetics in 2017. What they found when they measured the level of outside genetic influence on Nilotic populations was this: essentially none. The statistical test they used, an F3 test for gene flow, showed no significant signal of admixture with outside populations in the Nuer. For the Dinka, only a very minor signal appeared. The conclusion was stated plainly in the paper. Nilotic populations show long-term continuity, genetic isolation, and genetic links to ancestral East African people. And when they measured genetic differentiation between the Dinka, the Nuer, and the Shilluk, three distinct peoples with distinct languages and distinct histories, the FST value came back at 0.0028 or below. That is near zero differentiation. Three different peoples, practically the same genome. The isolation wasn't tribal, it was regional. The entire Upper Nile corridor maintained its genetic integrity together. This isn't just in the blood, it's in the economy. The Dinka cattle economy, the bride wealth system, in which marriages are formalized through the transfer of cattle between families, structurally reinforced marriage within the community for centuries. It was never designed as a genetic preservation strategy. It was a social and economic institution. But its effect, played out across hundreds of generations, was to keep the Dinka genome closed. A cultural institution became a genomic fact. This isn't just ancient history. This is in your DNA right now, or rather in theirs, intact, in a way that almost no other population on Earth can match. Consider this. In 1995, researchers measured height and body mass in multiple Nilotic groups living together in a refugee camp in Ethiopia. Same food supply, same shelter, same disease environment for everyone. And the Dinka and Nuer men were still significantly taller than Anuak and Shilluk men in the same camp. If you want a controlled experiment for genetic height inheritance, a refugee camp, where every environmental variable is equalized, is about as close as you get in real life. The Dinka still stood apart, under identical conditions, in a refugee camp. The genome doesn't lie. Actually, hold on. We talked in our last video about how a single migration wave out of the Eurasian steppe replaced most of Western Europe's ancestry in less than 5 generations. Now we're looking at the exact opposite, a population that held its genome together for 50,000 years. Which strikes you as the more powerful force in human history, the expansion that replaces or the resistance that refuses to be replaced? Genuinely curious? Drop it below and keep it in mind, because the next part of this story involves both. 50,000 years ago, the question wasn't where the Dinka were going. It was who was already there. In Kenya, at a place called Panga ya Siidi, a rock shelter above the Indian Ocean coast, an individual was buried 18,000 years before the present. One of 6 ancient genomes in Lipson and colleagues' 2022 Nature study. His genetic composition, when they ran the models, required 3 source populations to explain. One of those populations doesn't correspond to any living group. It appears in the data. It leaves a signal, and then it's gone, a ghost component, an ancestral signature that has no surviving population to claim it. Everything I just told you about this burial? Hold on to it, because it's about to mean something completely different. Because here's what else Lipson and colleagues found in that same study. The 3 anchor populations used to define the entire genetic space of their analysis were the San, the Mbuti, and the Dinka, which means the Dinka genome in a Harvard laboratory in 2022 contains at least a partial echo of that 18,000-year-old man's world. He is gone. The signal isn't. And then there's the 2019 science paper from Dr. Mary Prendergast at Washington University, working again with Dr. David Reich's team. Ancient DNA from the Pastoral Neolithic in Kenya, the people who introduced cattle herding to sub-Saharan Africa 5,000 years ago. When they modeled the ancestry of those early pastoralists, they found something that genuinely surprised the field. Approximately 50% of their ancestry traced back to a Dinka-related source. The other 50% came from ancient herder populations in the Levant, what is now broadly the Near East. These 2 streams of ancestry met in northeastern Africa and produced the people who walked south with their cattle and changed the entire genetic and cultural map of the continent. Here's the loop that closes. The Dinka's cattle economy preserved their genome. But that cattle economy, the cultural institution itself, traces back to ancestral populations who were already part Dinka in their ancestry 5,000 years ago. The practice came from the genome. The genome preserved the practice. It has been feeding itself for 5,000 years. There's a word in the Dinka language, more. It refers to the cattle camp, the seasonal movement of herds, the social and spiritual center of Dinka life. Today, there are Dinka men in Juba, in the Sud, and in diaspora communities in Minneapolis and London who carry that tradition, who carry Y haplogroup AM13, one of the oldest paternal lineages still present in any living human population, and who have never heard of a principal components analysis, and who are, nevertheless, in the methods section of a 2022 Nature paper, the fixed point against which the ancestry of an 18,000-year-old individual is triangulated. The ancestral population that built East African pastoralism is not quite the same as the modern Dinka. It's older, a diverged lineage from the same common source, a twin, not a parent, which means the Dinka are not the origin of this story. They are the only surviving branch of it. The source is the question that keeps pulling this story deeper. So let's close the loop on the body. In 2007, researchers built a climate chamber and did something that hadn't been done before. They tested Allen's rule directly, on living people, measuring actual heat loss rates in human subjects with different limb length ratios. What came back confirmed the principle. Longer lower limbs reduce the rate of mean body temperature increase during exertion in hot conditions, measurably, consistently. The Dinka somatotype, extremely lean, long distal limb segments, minimal body fat, sits at the theoretical optimum for thermoregulatory efficiency in environments above 35 degrees Celsius. The Upper Nile regularly exceeds 40. This is precision that looks like design. So here's the complete picture, assembled piece by piece. Ancient climate selected for a body type. That selection happened along the Upper Nile, in the same geographic corridor where the Dinka's ancestors were already distinct 50,000 years ago. The body type was preserved by endogamy. The endogamy was sustained by a cattle economy. The cattle economy produced the genomic isolation that makes the Dinka genome stable enough for modern scientists to use as a fixed reference point in studies of all of human prehistory. It closes, it fits, all the pieces locked together. Stay with me, because this next part is the reason this video exists. In 2015, the same year some of these ancient DNA studies were being finalized, a separate research team published their analysis of 232,000 individuals across 80 ethno-linguistic groups. We touched on this finding earlier. Here's the part we didn't finish. They didn't just find that genetic population structure explains 50 to 74% of body proportion variation. They found that genetic structure accounts for nearly all of the observed relationship between contemporary temperature and body proportions in both children and adults. The climate signal, the living response to heat, was almost entirely explained by genetics. Not by what the Dinka body is doing now, by what happened to the Dinka genome then. 3 seconds of silence here, because this matters. The Dinka man who runs across the Sud in 42-degree heat, moving efficiently, cooling himself through the geometry of his body, he is not performing a contemporary adaptation. He is not responding to his environment in real time. He is executing a genetic program that was written for his ancestors and then locked into the genome by the same endogamy that makes him useful as a scientific reference point. The thermoregulation is real. The mechanism that produced it finished running a very long time ago. What's happening in that body is ancient. What's happening in that genome is stranger still, because there is one thing about the Dinka genome that no model of pastoral adaptation predicts. They are among the world's most committed dairy-consuming pastoralists. They have been for thousands of years, and yet the mutation that allows adults to digest milk into adulthood, the lactase persistence variant that swept through European and East African pastoralist populations as cattle herding spread, that mutation is at low frequency or essentially absent in the Dinka. Researchers have confirmed this across multiple studies. The genome that preserved everything else somehow didn't acquire this one. And nobody fully understands why yet. What's happening in that genome is this. Harvard Medical School, Department of Genetics, 2019. Dr. David Reich's team and Dr. Mary Prendergast are running a QPADM analysis on 12 ancient individuals from Eastern Africa. QPADM, you need to know this term because it comes up constantly in this field, is a statistical framework for modeling ancestry. To run it, you need reference populations. Known stable genomes that define the genetic space in which the ancient individuals will be positioned. Without the references, the model has no coordinate system. You cannot locate anyone. The team selects 3 references. One is an ancient population from the Levant. One is a group of Stone Age foragers from Ethiopia. And one, the reference used to represent the deep East African genetic baseline, the ancestral Nilotic component, the oldest surviving signal of the population that was already in the Nile corridor, before the Pastoral Neolithic, before the Iron Age replacement wave, before any of it, is the Dinka. The lactase anomaly resolves here and into something larger. Researchers at Uppsala University, publishing in Human Molecular Genetics in 2021, confirmed that South Sudanese Nilotic groups, including the Dinka, have low frequencies or absence of common lactase persistence variants despite intensive dairy use. Their proposed explanation, the gut microbiome may be compensating where the genome is not. The Dinka genome is so intact, so old, so carefully preserved by centuries of endogamy that it appears to have found a biological workaround rather than acquiring the mutation that every other dairy-dependent population acquired. It routed around the update, a genome that old doesn't easily update. And now, here is what this video has been building toward. In Prendergast and colleagues' 2019 science paper, in Lipson and colleagues' 2022 Nature paper, in Wang and colleagues' 2020 Science Advances paper, 3 of the most important archaeogenomics papers published in the last decade, 3 of the most cited studies of human prehistoric migration and population movement in sub-Saharan Africa. All 3 published in the most prestigious scientific journals in the world. In all 3 papers, the living Dinka genome is used as one of the fixed reference coordinates to reconstruct the ancestry of all sub-Saharan Africans going back 50,000 years. The Dinka are not a subject of these papers, they are the instrument, not the thing being measured. The ruler, every ancient individual in those studies, every burial, every petrous bone, every fragmentary genome extracted from a 3,000-year-old Kenyan burial has its ancestry expressed in terms of its distance from, or similarity to, the Dinka. The Dinka do not appear in the conclusions, they appear in the methods. That is where coordinate systems live. That is what they are. You are not watching a documentary about the Dinka. You are watching a documentary about the measuring stick that was used to measure you. Dr. Sarah Tishkoff, one of the most respected geneticists working on African population diversity, has described populations like the Dinka as natural experiments in human biology. And they are. But the deeper experimental context is the one her 2009 science study helped establish. The Nilotic cluster, the Dinka, Nuer, and Shilluk, sits in a discrete genetic space, separate from every other African population, equidistant from several different major groups, belonging to none of them. That's not the position of a subject in an experiment. That's the position of a control group, the stable reference against which everything else is tested. The Dinka are the control. The rest of human history is the experiment. If it was never dissolved, the barrier, the genetic fault line that formed 2,500 years ago, then you have to ask what it looks like today. The Dinka are the carriers of some of the oldest surviving Y-DNA lineages in any living population. Haplogroup AM13, present in 62% of Dinka males, according to Hassan and colleagues' Y-chromosome study of Sudanese populations, is among the oldest Y-chromosome lineages still carried by living people anywhere on Earth. These are lineages that branched from the rest of the human Y-chromosome tree before the major migrations out of Africa, before Europe, before Central Asia, before the steppe, before all of it. And yet, the data said the exact opposite of what a simple story of ancient continuity would predict. The Dinka population underwent one of the most severe population catastrophes in the modern era. The Second Sudanese Civil War, 1983 to 2005, killed an estimated 2 million people, the majority from South Sudanese Nilotic communities. The conflict that erupted again in South Sudan in 2013 internally displaced over 4 million more. The population that carries the most ancient surviving paternal lineages in any living people, the reference coordinate against which 50,000 years of human prehistory is mapped, experienced mass displacement, mass death, and mass diaspora within a single generation. The 2017 PLOS Genetics study from Uppsala University and the University of Khartoum was explicit about this. Nilotic populations show long-term continuity and genetic links to ancestral East African people that are dependent on geographic position. The isolation that made the Dinka genome scientifically useful is the isolation of staying in one place, in one genetic tradition, for a very long time. Displacement scale that occurred in South Sudan doesn't merely disrupt a culture. It severs the geographic anchor that made the genetic isolation possible. The reference coordinate is moving. And then Wang and colleagues' 2020 Science Advances paper showed something that connects the ancient to the living in a way that is difficult to hold at arm's length. The genetic boundary between Nilotic-related and Bantu-related ancestry in Eastern Africa, the same boundary produced by the Iron Age replacement wave 2,500 years ago, maps onto the contested population and territorial borders of the current conflict with uncomfortable precision. The genomic map of ancient population replacement and the contemporary map of ongoing conflict are not two separate documents. They are the same document, printed at different scales. There are Dinka individuals in Minneapolis, Minnesota right now, the largest Dinka diaspora community outside South Sudan, carrying Y haplogroup AM13, carrying the oldest surviving paternal lineage in any living population, working night shifts, raising children who speak English with American accents. The coordinate is in diaspora. The genome is intact, the geography is gone. The fault line that formed 2,500 years ago was never dissolved. It has simply been given different names in different centuries. Pastoral Neolithic, Iron Age replacement, colonial partition, civil war. The genome and the news cycle are describing the same event at different time resolutions. The Nile moves north. It has moved north for millions of years. It carries sediment from the East African Rift Valley through Sudan and into the Mediterranean, building a delta that reaches into a sea that connects 3 continents. It doesn't know what it's carrying. It moves because water moves downhill, and this is the path. Every major archaeogenomic study of sub-Saharan human prehistory from the past decade has used a genome from a people who live beside this river as the fixed point from which everything else is measured. The river doesn't know that either. Lipson and colleagues' 2022 Nature study working backward from 18,000-year-old ancient genomes, using the Dinka as an anchor, found evidence of mating among widespread human groups beginning 50,000 years ago, followed by a shift toward regionalization about 20,000 years ago that produced the distinct genetic clusters we recognize today. The Dinka cluster diverged from the San cluster and the Mbuti cluster in the deep Pleistocene. Together, these three surviving lineages represent the oldest branches of a human genetic tree whose root predates the construction of any monument, any city, any written word by 100,000 years. Now here is the personal part, the part the data makes unavoidable. Dinka-related ancestry at 24 to 33% was present in ancient Kenyan populations between 2,300 and 3,500 years ago. Those populations contributed ancestry to Iron Age groups. Those Iron Age groups spread across Eastern, Central, and Southern Africa. If your ancestry includes any East African line, from any branch, at any depth, the probability that your genome carries a measurable echo of the ancestral Nilotic component, the same component that functions as a reference coordinate in the reconstruction of all of human prehistory, is not zero. You may carry the measuring stick. You may never know. If this changed the question you're asking, not just about the Dinka, but about your own genome, your own distance from the reference point, subscribe, because the next episode follows a different coordinate system to a completely different corner of the human story. The tallest people on Earth did not become the reference point for human history because of their height. They became the reference point because they stayed in one place, in one genetic tradition, in one relationship with their cattle and their river, long enough for the rest of humanity to diverge so far that someone needed a fixed point to navigate back. The Dinka are that point. Everything else, including you, is the distance from it.